Nitrate is a form of nitrogen and is a type of nutrient. There are other forms of nitrogen, such as ammonia, that we also refer to as nutrients, and that, when present in excess in aquatic systems, can lead to algal blooms. Nitrate specifically also has a drinking water limit of 10 milligrams per liter, meaning that drinking water containing more than that value is considered to be unsafe. Elevated levels of nitrate can be caused by fertilizer runoff, animal waste, sewage, or atmospheric deposition. Pictured here is the equipment that we use for nitrate, which is one of our more complicated methods. You'll notice that there's 2 pipettes, and we'll also be using a timer, alcohol wipes, and then that green box in the back middle contains vials and reagents that we'll be using. The large box in the top left is a spectrophotometer. When the reaction takes place in the vial, the liquid within the vial will change color based on the quantity of nitrate present. Meaning, with a large concentration of nitrate, the color change will be more dramatic than only with a slight concentration of nitrate. The spectrophotometer uses this change in color to determine how much nitrate is present within the sample. For nitrate, the process starts with unopened vials that already contain a small amount of acid. We pre-label these vials to make sure that their numbering matches the numbers of the samples that will come in later to prevent any mix-ups during the process. The first step, once you've opened the vial, is to pipette a small amount of sample into the vial. Here, the sample will mix with the acid and alter its pH, which is important for later steps in the process. For Stream Team, we'll often do this step for 10 to 15 vials before we move on to the next part of the process. Once you've pipetted sample into all of the vials in your batch, the next step is to add a liquid reagent. That small bottle with the green cap is what has the reagent for nitrate. You'll be using a different pipette and different pipette tips. With this reagent, you'll want to keep the cap on when you're not pipetting to make sure that you're not letting any of the fumes out. Some of our reagents are light sensitive. With this one, it simply emits a noticeable smell. And again, you'll notice that I remove pipette tips between samples to assure that there's no cross-contamination if I've touched the bottle with the end of the pipette tip. Once you've finished pipetting both, you'll then invert 3 times to make sure that they're thoroughly mixed. Now that the reagent has been added, we will wait 15 minutes for the reaction to take place. While that's happening, we will be cleaning the outside of the vials. This is important to do because the samples are read by passing light through the vial, and any flaws in the glass or anything resting on the outside of the glass can affect the way that the samples are read. So we use these alcohol swabs to make sure that the glass is totally clean before we read them. Now that the vials are totally clean, the only thing left to do is read them. You'll take them and invert them once more, just to make sure that they're thoroughly mixed before you put them into the spec to read them. You put it into the slot and then cover it so that no extra light can get through, again because it's using light to read the sample. You'll hit the scan button and give it a second to read the sample. It will give you back a number, and you will record that as well as whether the number is negative or under range, and then you're done.